This is my great pleasure that BioTuring hosts the webinar, and our special guest today is Dr. Ankur Sharma. Ankur is a scientist at the Genome Institute of Singapore and the Cancer Therapeutics and Stratified Oncology. So at his work, he combines experimental and computational approaches to study human tumors as a complex ecosystem, and also its evolution under various selection pressure. In particular, he tracks the evolutions of tumor and their ecosystem in individual breast cancer patients before and after drug treatment, one cell at a time, using single-cell technology. And recently, he published the papers on Nature Communication in his, that study the longitudinal single-cell RNA-seq in drug-resistant cancers. And now it is my great pleasure introducing Dr. Ankur Sharma.
So thanks for the introduction. And so today I'll be talking about a recent work where we're trying to look at tumor evolution under the selection pressure of chemotherapeutic drug, and how we use single-cell genomics to understand how a tumor cell will behave when it is given a challenge of chemotherapy. So the question which inspired us is that, or the problem which inspired us is that, why some patients respond to chemotherapy but others not? So in any given group of patients, we will find two types of patients, one which respond to the chemotherapy and they are responders and they do pretty well. But then there are some patients which does not respond to the chemotherapy. And mostly these patients, we call them non-responders. And there are various reasons why these cells does not respond to the chemotherapy. And some mechanisms have been discussed, like cancer stem cell, epithelial to mesenchymal transition, or sometimes these cells are dormant, they are not proliferating. But nonetheless, these patients does not respond to chemotherapy, which lead to the tumor relapse, and in some cases, death of these cancer patients. So this is the area which we are really focused on, like why this and how these cancer cells adapt a mechanism to overcome the chemotherapy. And when we think about tumor, and it has all the evolutionary mechanism which a human population use. For example, if you look at the 4 major principles of Darwin, which are variation, inheritance, high rate of population growth, and differential survival and reproduction. So tumor cells follow all these things, and this differential survival, which lead to the survival of some cells under the chemotherapy and other cells does not survive in chemotherapeutic pressure. So looking at the tumor as an evolutionary system is now almost 40-year-old concept, where first time E.C. Nowell proposed this idea that tumors are a clonal population, and in one tumor, there are actually multiple clones. So when we think of a tumor, we think of one tumor, but it is actually multiple tumors, and each tumor in its own is an entity which responds differently to different kind of selection pressure. And I think this concept gained much more prominence in this 2012, after this 2012 paper from Charlie Swenston, when they were looking at the different part of the tumor, and what they found is that different part of the tumor have different kind of mutation, which means now these cells have different clonal fitness, if we say so. And so what happens in a tumor if we think, let's say, assume there are multiple clones, and in this case, I'm showing there are 5 different clones, and a selection pressure may lead to elimination of most of the clones, which will lead to the tumor regression. And in clinic, or in some time, we think that tumor is responding to the chemotherapy. But in some cases, there could be few clones, as in this case, I'm showing clone 1 and clone 2, which does not respond to the chemotherapy, right? But it leads to the tumor shrinkage, and as soon as when we take out the drug, now these clones start proliferating again, and which give rise to the tumor relapse. So what we understand about this entire process, so we know how, when a patient comes to the clinic, how a tumor look like. So a tumor is a mass of these heterogeneous cells, and when treatment start, as I mentioned, most of these cells will die, but in some cases, some cells may survive. And when a tumor shrink significantly, these patients will be given drug holiday, because chemotherapy is highly toxic, not only to cancer cell, but to the rest of the cells in the body. And when the selection pressure is taken away, now these residual cells may start regrowing and will lead to the tumor relapse. So from the clinical data, we know how a tumor look at the presentation, right? We also know how a tumor look at the relapse, because in both the cases, there is some kind of surgery or biopsies involved. But what we don't understand, how this process is happening dynamically, like how when a patient is undergoing chemotherapy in real time, what happens. And it is very difficult to understand this in clinic, because most of the time we cannot collect the patient sample when it is undergoing the chemotherapy in the clinic, right? So we wanted to model this part, because this part where least amount of information is there. And we thought there could be two possibilities how these tumor cells acquire drug resistance. One, as I mentioned, there could be some residual cells which survive the chemotherapy and give to the entire tumor, give rise to the entire tumor. In another possibility, there could be a scenario where you have tumor cells which to begin with are not resistant, but when a selection pressure is applied, these cells somehow reprogram themselves by the means of epigenomic or acquiring new mutations. And now this lead to the generation of a new population which is resistant to the chemotherapy. So this could be acquired by genetic or phenotypic plasticity. So we wanted to understand which one of these is happening in the context of the oral cancer cells. So in our lab, we are generating models of patient-derived cells. So we collect the tumor specimen from the patient and grow these tumor cells in the lab. So what we wanted to understand in this case, we started profiling our tumor cell lines, and we found that there are two different kinds of cell lines which were very interesting. One cell line which is highly epithelial to begin with, so we call it phenotypically homogeneous cells. As you can see, most of these cells are expressing E-cadherin, so these cells are highly epithelial in nature. And we chose another model which is where you have both epithelial, which is green, and mesenchymal red cell line. Now this is important because we wanted to understand how the phenotypic heterogeneity impacts the tumor evolution. So in one case, we are looking at phenotypically homogeneous tumor. In another case, we are looking at phenotypically heterogeneous tumors. And the way we did this experiments is that we took 100 cells from each of these culture and we plated them in 24 replicates. And each replicate is given a same amount of the drug, which is IC50 for the standard of care drug cisplatin. So why we did this experiment, why we had 24 replicate? Because we wanted to understand, is it a stochastic process or a deterministic process? And our readout would be, if this is a stochastic process, right? So if there are different mutations are acquired in different cells, then you will see out of 24 clones or out of the 24 replicates, some replicates will give rise to a drug-resistant clone, others will not. While if it is a deterministic process, then same mechanism will happen in all of these cells and most of the wells will give rise to the resistant clones. So this was one of the idea of behind doing this 24 well plate experiment was, and then what we found is that every time when we seed 100 cells, we found same kind of drug resistant properties emerging in all the wells. So which give us two indications. First, the drug resistant population is present in 1 in 100. So like even the 100 cells can give rise to a drug resistant clone. Also, this is a very deterministic process because every time in each well, we found the same kind of phenotypic switch. So I'll show you the example of one of the, like how when we did the screen, how this looks like. So in one of the well, when we look into this well at the beginning of the experiment, you see there are two different kinds of cells, green, which are epithelial, and red, which are mesenchymal. And drug selection led to the elimination of both the clones because as I mentioned, most of the cells will die in the drug treatment. But few green cells and few red cells survive within first 2 weeks of the treatment. When we continued this treatment, now you see these green cells started growing bigger in colony, and that there are few red cells which are still surviving and trying to cope up in the selection pressure of the drug. But after 6 weeks of the treatment, we do not see any red cells. So which means all the mesenchymal cells were dead, but and all this plate is dominated by these big colonies of the epithelial cells as shown in the green. So suggesting that a green cell started from time 0, dominated the entire plate after 6 weeks of the culture. So this is a classical example of Darwinian selection when there is in the pool of multiple clones, there is one clone which is fit and give rise to the entire colony. Another example where we saw that in a group of homogeneous cells. So as you can see, most of these cells are homogeneous. They are expressing this green E-cadherin protein. Within 2 weeks of the selection, we started seeing that this de novo emergence of these red cells. As you can see now, these cells are both red and green, suggesting there's a phenotypic plasticity happening in these cells. They express now both epithelial and mesenchymal markers, suggesting their transition towards EMT. And when we kept these cells in the drug for a long time, now these red cells kept on proliferating more and more. And at the end of the experiment, now we have a culture which expresses both epithelial and mesenchymal marker. So suggesting in this case, it starts with a very sensitive epithelial population, but evolve or adapt itself into a epithelial mesenchymal phenotype. Now, this is an example of adaptation where, to begin with, these cells do not have all the requirement to overcome the drug treatment, but they adapt themselves and gain these new requirements. So we found all these things is happening in plastic, which is very good. But as we understand, the plastic, or like when we culture cells in a Petri plate, it does not have an environment. So we wanted to address the question: if we take all this and this entire setting into in vivo system, where you have endothelial cells, stromal cells, how this will impact the tumor evolution. So now what we did, we took both of these cell types, injected them into the mouse, made the PDX model. And on this PDX model, when tumor was full grown, we started injecting this chemotherapy drug cystatin again. And we did this in vivo evolutionary model. So when these cells were, when these tumors were treated for almost 2 months with this drug, and we harvested the tumor in 2 groups, one which was treated with the drug, another which were not treated with the drug. So what we found is that before drug treatment, these one of the models, which is we call 120, has epithelial cells, but they lack the mesenchymal markers in vimentin. While tumor evolution, what happened? They lost their epithelial property. As you can see, there's a reduction in the E-cadherin population, and there's a de novo appearance of this vimentin positive cell. So which goes in the same direction what we showed here. To begin with, these are epithelial, but they become mesenchymal as tumor evolution is going. In another model, which is 137, we have both epithelial and mesenchymal cells, but drug treatment led to the selection of these epithelial cells and complete elimination of the mesenchymal. Again, as we showed in the in vitro model, you have both epithelial and mesenchymal cells, and drug selection completely eliminate the mesenchymal cells. Finally, in PDX model also, we understand the immune system is not as competent as is in human, and the amount of the drug is given to this patient might not be physiological, right? So we wanted to understand what happened to these tumors when they were treated with this drug in the clinic, right? So as I mentioned, we were making these patient-derived models. So when we were collecting these cells from the patient was going with the same drug because cisplatin is standard of care drug. So patient was undergoing with the same treatment in the clinic. So when we obtained the tumor specimen before drug treatment and after drug treatment, we found that there was increase in the mesenchymal property in the case of 120, and there's an increase in the epithelial properties in case of 137. Again, suggesting that in 3 different evolutionary paradigms, in plate, in PDX, and in clinic, we found the similar observation. While there's a phenotypic plasticity in a model which is to begin with epithelial, and there's a selection of the tumor cells in a more heterogeneous model. So now we wanted to understand what is the molecular mechanism of this. Like in this group of epithelial and mesenchymal cells, when few epithelial cells survive, like what is the frequency of these cells? And when we have this group of phenotypically homogeneous cells, which were green in begin with, and they start expressing vimentin, is there a small population of vimentin expressing cells which is pre-existing? So in short, like are there rare population which were pre-existing and we can find them? And as most of you who work with these cellular population, you appreciate that it is very difficult to identify these rare population. It's like finding Waldo in this pool of people. So our task was how to find this wild Waldo, and or the rogue Waldo, and what we adopted the single cell RNA sequencing approach, because in this you have advantage of looking at individual tumor cells, one cell at a time, as we say, and to ask the question, what was the phenotype of the cell before drug treatment and what happened to this cell after drug treatment. So you can look at the cell longitudinally, right? So we did this experiment. And so in total, we had 700 cells which were naive, like they were untreated with the drug, coming from 3 different patient, primary and metastatic size. We made 4 different drug-resistant models. We had drug holiday models because, as I mentioned earlier in the clinic, and a patient at the presentation, during, and after drug treatment. So, and we did the single-cell sequencing with these 1,300 cells. So in total, we had 3 patients, 13 cell type, 4 evolutionary time points. And when we cluster these cells, we found there are 5 different clusters, but 4 of these clusters are the major cluster, as you can see in this Disney plot and this heat map. And when we look at the gene expression properties of these cells, we found that they beautifully group into 2 different kind of developmental trajectory. One, either they are basal or epithelial, so which is cluster 2, or they are luminal or mesenchymal. So two different cell states, like, or cell types, epithelial, mesenchymal, basal, and luminal, can easily cluster this data into 2 groups. Now, when we started looking at the individual tumor population, so as I mentioned, the tumor which is epithelial to begin with, cluster here in epithelial group. During drug treatment, these cells completely change their transcriptome from epithelial to mesenchymal. So it was interesting that we could not find any mesenchymal population, suggesting it's an example of plasticity, not example of selection. So which means all the cells adapt from epithelial to mesenchymal state. In another case where there were 2 different kind of populations were present, so some cells were epithelial, other cells were mesenchymal, and drug selection lead to the elimination of the mesenchymal cells, which was very interesting because in literature there are examples. In some cases, mesenchymal cells are more resistant, but surprisingly, we found that these epithelial cells were more resistant in our case. And this is an example of selection where you have both epithelial and mesenchymal, or 2 different clones were present, but only 1 clone got selected. Now, the interesting part is that both the clones are coming from the same group, like both the clones to begin with are epithelial in the nature. And we asked the question, why if both the cells are starting from an epithelial state, right, both two groups, why one took a more epithelial route while another took a more mesenchymal route? So why the same cell type took 2 different divergent paths, if we see so, to acquire the drug resistant? Is the answer lies in the type of the cells, or like cell of origin in this case? So for that, we look at the stem cell potential of these cells, and we cluster these entire 1,300 cells based on the different stem cell markers. And we found this remarkable heterogeneity in the stem potential of the epithelial group, while mesenchymal cells relatively were more homogeneous in their stem potential. Now, this was kind of expected because these are epithelial tumors, and you would expect the epithelial cells to show more stem potential, while a mesenchymal cell type is more differentiated. So once they become mesenchymal, they are already differentiated. In this epithelial group, we also identified they express SOX2. Now, the SOX2 is very important here because SOX2 is a known oncogene and tumor initiating factor in squamous cell carcinoma. It was very interesting to see the high SOX2 expression in this epithelial cluster, which is high stem-like. But what was also surprising to see that all these mesenchymal cells lacked SOX2. So that was very interesting that once it's becoming mesenchymal, they are somehow losing SOX2. So as you can see in this graph, to begin with, these cells, primary cells or naive cells, have SOX2 expression, but during drug treatment or metastasis, these cells completely lose the SOX2. While in another model where epithelial cells were getting selected during drug resistant, so you see in the sensitive cells, we have SOX2 expression, drug resistant cells retain this SOX2 expression, while the metastatic clone lost it. So which means when cell was maintaining its epithelial property, it retained SOX2, but when cells transitioning from epithelial to mesenchymal fate, it lost SOX2. So it was kind of interesting, but also surprising. So we wanted to ask the question, if cell is losing SOX2, is it still maintaining its stem potential? Because if SOX2 is so much important for stemness, why a drug-resistant cell will in first place lose it? right? So we did this limiting dilution tumor experiment where we had both SOX2 and SOX2 positive and SOX2 negative cells, and we injected these cells in different dilution. And it was interesting to see that even the 1,000 cells, irrespective of their SOX2 status, were able to form the tumor. Now, this was kind of puzzling. These cells lost the SOX2 expression, but retained their stem potential, become more drug resistant, became more metastatic. Now, so what's happening to the stem potential? How these cells are retaining their stem potential in the absence of SOX2? So at this point of time, we hypothesized there might be a stem cell switch happening because you don't have SOX2, but you somehow need to maintain the stemness. So what is this stem cell switch? That's what we wanted to ask. But before that, we wanted to test this. Is there a really stem cell switch happening, and is it SOX2 dependent, or are we looking at the real marker or not? So with this, we did this stem ID analysis, and we look at the different stem cell state of these two groups, and we found that indeed there was a switch in stem cell state from primary to the drug resistant cells in this model of 120, while 137 they retain their stem state. So which again goes very well with the SOX2 thing, that in 120 there's a loss of SOX2 and there's a change in stem state, while in 137 there's a retention of SOX2 and maintenance of the stem state. So we did this phenotypic screen for 2,000 transcription factors to identify what is the master regulator of this stem cell switch, because if there's stem cell switch happening, it might be a transcription factor which is replacing SOX2 and doing all the function which SOX2 do for stem cell maintenance and all. Surprisingly, we found SOX9, and which was very interesting because SOX2 and SOX9 are both coming from the same family. And with this data suggests that loss of SOX2, in the case of loss of SOX2, now SOX9 rose to the occasion and started replacing SOX2 as a stem cell factor. Indeed, when we dig deeper into our data, we found that at the single cell level also, there's an increase in SOX9 expression from sensitive to the resistant cells. And that is true for from the primary to the metastatic also. So what we found here is that during tumor evolution, these cells lost SOX2 and gained SOX9, and now they're dependent on SOX9 for maintaining their stemness. So we did another analysis, and that's the beauty of doing single cell. You can ask multiple questions. So in this case, we did this analysis. We're looking at, is there any SOX2 positive cell which is expressing a mesenchymal marker vimentin? And as you can see in this plot, there's a complete anti-correlation. So we could not find any SOX2 positive cell which shows mesenchymal property. Again, goes very well with all our observation that SOX2 is mainly expressed in epithelial cells, and all the mesenchymal cells lose SOX2. Also, when we look at the SOX9 and vimentin expression, we found a very good concordance. So most of the cells which were expressing SOX9 were also expressing vimentin, suggesting SOX2 is important for epithelial fate, while SOX9 is important for mesenchymal fate. And then we did this, when we look at the SOX2 and SOX9 expression in some of these models, so as you can see, these cells, to begin with, they have lower level of SOX2, and during tumor evolution, they gain lower level of SOX9, and during tumor evolution, they gain SOX9 and they gain the vimentin. So it goes well with this thing. And when these cells lose SOX2, or SOX9, so all the drug resistant cells now they acquired, or like they started using SOX9 as a stem factor. So when these cells lose SOX9, they don't like it and they start dying. So suggesting that SOX2 is important for epithelial cells, SOX9 is important for mesenchymal cell, and SOX9 is maintaining the stem potential of drug resistant cells. So far what I have shown you is that there's a transdifferentiation happening during tumor evolution, epithelial cells becoming mesenchymal. There's a complete change in the transcriptome of these cells from epithelial to mesenchymal, and now they change their allegiance from SOX2 to SOX9. The question comes is that if there's this change in stem cell factor and the entire change in the transcriptome, how the stem cell factor is helping or leading to this change in transcriptional reprogramming? Is it via epigenetic factors? Because SOX9 is a known pioneer factor. So we ask the question, if the de novo role of, or like this new role of SOX9 is somehow linked to the epigenetic regulation. So for this case, we first bin our data into two groups, one which is genes which are highly expressed in sensitive cells and genes which are highly expressed in the resistant cells. And most of these genes were epithelial to mesenchymal transition related genes. And now we did a ChIP-seq for H3K4ME3 mark, and when we specifically look at the genes which were highly expressed in the drug-resistant cells, and we look at the promoter of these genes, surprisingly we find that these promoters showed open signature, which was surprising because this gene is not expressed in a sensitive cell. Then why this promoter is already marked by K4ME3, which is a signal of an open chromatin? We asked the question, is this active chromatin or not? So when we look at the activity of these promoters, we found that in H3K4 K27 acetylation chip, these promoters were poised or like lacked the acetylation mark in the sensitive cell, and during tumor evolution, they gained the acetylation. So this is suggesting that they were kind of in a poised state and now gained the activation when these cells started, culturing in the drug, right? So more interestingly, we asked the question, is this a bivalent promoter? So for some of these promoters, we did H3K27 ME3 chip, and we found that, as you see, during tumor evolution, there's a gain of the acetylation and lack of the K27 ME3. So now they lose repressive mark and they gain the activation mark. So these are some of the example of the EMT promoters. As you can see, in the sensitive cells, they already are in open state, but they are not active. And during tumor evolution, they gain the activity. And same is true for vimentin, interleukin-6, and Gas6. So to cut the long story short, what we found that these promoters are already in a poised state. They gain the transcriptional activity during tumor evolution by virtue of gaining the acetylation at this promoter and by losing this repressive mark. So which means like these cells are poised for resistant, and the selection pressure led to the activation of this chemo-resistance. So when we look at what are the transcription factors which were like differentially or like transcription factor motifs which were differentially enriched in the peaks in the sensitive versus resistant cells, not surprised, like as we expected, we found that SOX2 is more enriched on the sensitive enhancer in the sensitive cell, while SOX9 is more enriched on the enhancer of the resistant cell. So now this is again reconfirming this stem cell switch, saying that even at the chromatin level, we found this signature that there is a change in from SOX2 to SOX9. We went back to the patient tumor. As I mentioned, all these things is happening in patient-derived cells. So we can go back to the patient tumor and validate our data. And when we are looking at these patient tumor, you see, like to begin with, in one of the model, there are two different kind of cells, both epithelial and mesenchymal, shown in green and red here. And this tumor has both SOX2 and SOX9. SOX2 is in green and SOX9 is red. But during tumor evolution, as you see, these cells lost completely the mesenchymal expression. Now you see these are epithelial colonies in the green, and these cells also enrich the SOX2 expression in the tumor. So again, whatever we were observing in this in the patient-derived cells was actually true in the patient tumor itself. In another model where there was patient tumor is highly epithelial, as you can see in the green, and when we look at the stem cell compartment of these cells, they express both SOX2, which is in green, and SOX9, which is in red. So now the SOX2 expressing cells are very low here, which is not surprising because SOX2 is a cancer stem cell marker, and you don't expect too many stem cells in a tumor. But interestingly, when we look at this tumor, after evolution, as you can see that now they start showing this epithelial to mesenchymal transition like a feature. It lacks ecadherin expression and start de novo expressing vimentin in the tumor itself, and it completely stop expressing the SOX9 and change itself from SOX2 dependent tumor to a SOX9 dependent tumor. So again, revalidating, recapitulating what we see in the patient-derived cell lines. Now, so this is the example of the two patient-derived models. So we've asked the question, is it really happens in a multiple group of the patient? For this, we collaborated with, we collaborated with the Australian group, which had this rare access of this patient tumor before and after drug treatment. So in this group of 28 patients, we look at the tumors and we asked the question, how many of these show this plasticity versus selection phenotype? And as you can see here, in 6 out of 28 patients, we observed this example of plasticity where tumors begin with is epithelial and SOX2 dependent and become mesenchymal during chemotherapy treatment and start expressing SOX9 and completely lack the expression of the SOX2. While 22 out of 28 patients show this mixed tumor, which is SOX2, SOX9 positive and epithelial to begin with, and retain or gain epithelial properties during tumor evolution and enrich for SOX2. So we found in at least 80% of the tumor, they follow Darwinian principle of the tumor evolution, where they either increase their epithelial or mesenchymal property during tumor evolution, while 20% patients show this example of phenotypic plasticity, which might be acquired by epigenetic switch. So this is our working model, and what we propose here is that to begin within epithelial cells express both SOX2 and low level of SOX9, and this allows cells to maintain or a poised chromatin on the mesenchymal promoter. And as cells start losing SOX2 and gaining SOX9, which lead to the histone acetylation of K27 acetylation of these promoter, lead to the gain in the transcriptional activity of these cells. So we asked the question, if there's a gain of the acetylation which lead to the resistance, can we reverse this by blocking the acetylation of these cells? And for this, we did a screen for 150 chromatin remodelers, and we found BRD4 as one of the key molecule which inhibits the acetylation of these promoters and lead to the and change the resistant phenotype to a sensitive phenotype. In a complementary small molecule screen, we found JQ1. JQ1 is a small molecule inhibitor of the BRD4, so this fits very well. And BRD4 itself is a histone acetyltransferase. So which makes sense when we inhibit a histone acetyltransferase. Now there is no more K27 acetylation. These cell cannot evolve themselves into a resistant state and become more sensitive. So this is exactly what we see. Before JQ1 treatment, these cells are highly resistant to the cisplatin treatment. So this is the percent inhibition, and the presence of cisplatin, there is no inhibition. But JQ1 alone or in combination with cisplatin can completely reverse this cisplatin resistant to the sensitivity by inhibiting the acetylation of these promoters. So this is our working model, and what we propose here is that in a phenotypically heterogeneous tumor, these cells might be in a poised state where application of any selection pressure, it could be chemotherapeutic pressure or stress during metastasis where cell has to identify a new niche, may lead to this phenotypic plasticity of these cells, which is helped by change in stem cell factor from SOX2 to SOX9. And this allows these cells to become from epithelial to mesenchymal and allow them to adapt well in these hostile condition of the chemotherapeutic treatment. So this is we call a camouflage kind of model where cells to begin with look like epithelial but with a poised chromatin for mesenchymal. In another model where we see a phenotypic heterogeneity, we call this a division of labor kind of model where you have different kind of tumor population and each with its own function. So for example, in this heterogeneous tumor, there are some cells which are slow cycling and which might lead to the resistant for the a drug which kills fast dividing cell, because if the cell is not dividing that fast, a drug cannot kill it. Or there will be some cells which are already expressing these mesenchymal marks and which allow it to settle well into a distant metastatic niche. So this is what these are the two different models. One is where a cell show a plasticity, another where there's a Darwinian selection happening. And we recently published these results in this Nature Communication paper. So this is our model, and now what we are planning to take this model further into different patient tumor, for example, looking at the breast cancer or the leukemia, and as and when patient undergo chemotherapy in the clinic, and will be very interesting to see how tumor evolved under the selection pressure of this drug. So I did this work in the lab of Professor Ramanuj Dasgupta at the GIS, and I'm very thankful to Ram for his kind support and mentorship he provided me in the last 3.5 years. I'm also very thankful to the Elaine, who was a brilliant computational tech in our lab, who helped in some of this data analysis, and our collaborator Vibhor, who was helping during ChIP-seq analysis, and all our clinical collaborators, specifically Professor Gopal, Pierce, Floron, and Elaine. So thank you, and I'll take questions if there are any.
All right. So thank you very much, Ankur. And any questions, please send as a text message to BioTuring team so that we will coordinate the question. So we got some questions here. Can you help me?
Yeah. So one question is that did you observe any transdifferentiation happens in the heterogeneous population? So you're comparing the two models, the homogeneous and the heterogeneous one. So do you also observe the differentiation of the tumor cells in the heterogeneous population?
Yeah, I think it's a very fantastic question. So when we look at this heterogeneous population, and we could not identify any signature of the transdifferentiation. And what I think is happening is because there is already a population which is slow cycling. So when we added this drug in heterogeneous model, all these E-cadherin positive cells were the label retaining cells, and I think they survived fast, and that's how they led to the colony. But I assume if there is no E-cadherin positive cells, there might be a sign of transdifferentiation, but we have not done that experiment. Did I answer your question?
The next question is that we saw a very different response in the way the tumor cell gaining their resistance in the two models. So is that something to do with the clinical effects of these two groups? Like they become, one group will become more aggressive than the other, or they just gain the resistance?
Yeah, so I think if I understood your question correctly, you are asking if why two different groups showed two different kind of strategies for evolution. Is that correct?
Yes, that's right.
Yeah. So I think so we think we understand the mechanism partially, and that is by in some group of patients there's a heterogeneity and which allow them to have this different kind of cell population, which allow them to select under various selection pressure. But not all the tumor are equally heterogeneous. Classically, we thought that tumor which are more homogeneous, they are easy to treat, but our data suggests otherwise. What our data suggests is that even if a tumor is heterogeneous, it might not be that easy to treat it because it might have some of these covert mechanism like these epigenetic changes, which may lead to the tumor evolution. So what we think going forward we should be doing, traditionally we have looked at the mutations as a biomarker for drug response. And these days, like currently we are going towards a gene expression as a biomarker for the drug response. I think we need to combine all these technology like genomics, transcriptomics, and epigenomics, because epigenome is a layer which will lead to the transcriptional change. And many a time we miss this layer. We just look at the genome and transcriptome without giving too much importance to this epigenomic layer, which I think more and more people are coming suggesting that epigenome is very important, and it can be in a poised state. So it's not very easy to look for epigenome phenotypically, and that's why you need to dig deeper into the epigenome. Did I answer your question?
Yeah, that's okay. But there's one more thing that I saw in the homogeneous group, when they gain the resistance, those resistant cells are they look very similar to the metastatic cells. So is that suggesting that if you use chemotherapy on the tumor that is more homogeneous, then it will more likely to create metastatic?
Yeah, it's a fabulous question. Yes, you are correct. So what our data suggests that in these kind of tumor, and I showed you 6 examples of out of 28 in clinical specimen also. So these tumors were to begin with more epithelial, and when the chemotherapy or cisplatin was applied to them, these tumors become more mesenchymal. Now, this is very concerning. We go to clinic to get a chemotherapy so that it eliminates the tumor. But in some cases, what we are finding is that chemotherapy is actually aggravating the disease, changing a disease from an epithelial to mesenchymal nature, which is a poor prognosis. And when we look at the TCGA data, And when we look at the signature where patient have high SOX9 expression, low SOX2 expression, these EMT kind of genes, we found that these patients fare poorly during, like they show poor prognosis. So I think it's very important, and which is leading to a new concept of managing tumor, not trying to completely kill the tumor cell, because if a tumor cell can evolve, it will evolve into any selection pressure. It's just a matter of the time. So another thing is, can we manage tumor as a chronic disease? And there's lots of discussion going on in this area where we may not try to kill the tumor cell completely, but just like give the enough amount of the chemotherapy so they don't divide further. So all these are, I think, very interesting and emerging field in the cancer research.
Yes, yes. Yeah, it's really amazing. Okay, thank you.
Thank you.
Any more questions? So I think if we are not going to have any more questions, so I would like to thank Dr. Ankur Sharma for the excellent talk with his colleagues. And to the next part of the talk, let me introduce a little bit about the BioTuring Browser. So one current very challenging problem with the field is that the sequencing data, when being published in papers, theoretically, they are open access and accessible for every scientist. But practically, there are a lot of technical barriers that prevent the scientists either from academic labs or research companies or pharmaceutical companies that can access the data, and also to enhance the transparency and reproducibility of the papers. We develop BioTuring Browser so that scientists, as soon as a paper is published, scientists, medical doctors, the one without coding background can have all the state-of-the-art analyticals, algorithms, and data visualizations in order to reanalyze the results that the authors published and reported in the manuscript, as well as asking new questions from the data. I believe that this one can help, increasing the level of transparency in science. So let me give a quick, very quick demo for the BioTuring Browser. And, let me share the screen. So what is BioTuring Browser? So BioTuring Browser, we have multiple add-ons. One add-on is particular for single-cell dataset. So with BioTuring Browser, you can just simply use your laptop to view millions of data points of cells and this is actually the first software that can perform 3D notations on millions of single cells. And it has real-time tissue cell type prediction, and it has many more state-of-the-art and analytical functions. And the first questions that people ask is how to access the data from the paper. For example, the paper that Dr. Ankur just presented. So let's go to the browser and let's search for chemotherapy. And now you can see immediately on in the BioTuring Browser, this is the title and this is the abstract of the paper from Dr. Sharma and his colleagues. In difference from any other ways where you have to go to the Geo or short read archive to download the sequence or the expression data, here you just click Explore and all the data are now presented in the iterative dashboard. So on the left is the scatter plot of all the cells from 3 different patients that you probably have seen in this talk. On the right is the control panel. Now let's try to verify first questions. Now, whether, here we have 3 patients, 148, 137, and 120. And the P stand for primary, samples or cells from the primary site. And the M stand for metastasis. The PCR stand for the cells from the primary with drug resistance. And the PCR DH stand for drug holidays. So after some time, not without treating with the drugs. Now the first question is, let's try to look at the patient 137 primary, right? And the question is whether we have heterogeneous population. So in the papers, there are 2 cell types. One is epithelial and the second is mesenchymal cells. So the cell, how to identify the epithelial? We can use the EPC, the gene called EPCAM as the gene marker, and to see, okay, so this gene, which is the gene marker for epithelial cell, expressed here. The second gene marker is for the mesenchymal cell. Now we can enter the gene marker as bimentin, BIM for mesenchymal cell, and see. Now you see the red one, the red cells, it means that express EPCAM, and the blue one, it means it express the gene BIM, bimentin. And what are the yellow cells? The yellow cells are the cells that express both genes. So clearly that, we verified that the papers already correctly said, stated that for 137, it is heterogeneous populations. Now, what is interesting is that after treating with drugs, what happened is that the paper said that one cell type disappeared. And let's choose one after treating with the drug. This is primary drug resistant. Let's choose this one. And let's choose the drug holidays one as well. Now, again, let's try to verify the gene markers for both epithelial cells and mesenchymal cell, which is EPCAM and BIM. Now, clearly that indifferent from the previous primary cell before treating with drugs, now we just see that the EPCAM gene express and the VIM gene does not express at all. So this confirmed what the authors has stated. Now we move to another patient, which is 120. That's 120. 120 is here. Now in the papers, 120 is homogeneous population with just one cell type. And let's check. The same with the gene marker for epithelial cell, EPCM. Okay, it expressed. How about the bimentin genes? Does it express as well? If it also express, then this population is not a homogeneous population, but indeed it can contain 2 cell types or even multiple cell types. Let's check with the VIM. So clearly that VIM does not express in this population. So which means that there's a reprogramming from the one cell type, epithelial to mesenchymal cell. Oh, right. So, and confirm that the population is homogeneous. Now, the authors actually was trying to explain about this reprogramming mechanism and explain that it is because the SOX2 gene, the genes for the SOX2 genes does not express. And we choose any other. And 120, this one. And let's search for the gene SOX2. Now, you see on the left side is which one is on the left side? The left side. Okay. Now, let me see. And this is we are searching for SOX2. And SOX2 does not express in these populations, right? Which verify what the author has explained. And now what should we do? We can further ask a question. Okay, what is the gene expression differences before treating with drugs and after treating with drugs? So now we are looking at the two populations before treating with drug and after treating with drugs. And what we can do is we find the differential gene expression in these two conditions. Now we find differential expression analysis and drug differential analysis, and this is the result. Let's take a look at the result. Now, one population is before treating with drugs, and the second population is after treating with drugs. We can see it in the split view, so that on the top side, you can see the cells before treating with drugs, and on the bottom side, it is the population of cells after treating with drugs. Now, these are the set of differentially expressed genes. And let's take an interesting gene that the papers reported, which is vimentin gene. And let's see where it stays. Now, the vimentin gene here clearly it almost doesn't express in the population before treating with drugs for this population, and after treating with drugs, it appears. All right. So, and what we can see here is that this is the vimentin gene. And if you can annotate the vimentin gene here as well. And I think this is a very quick introduction of the BioTuring browser, and very quickly a user like myself can very easily verify, okay, we are very confident at the result reported with this paper, and this is why we invite the first author of this paper to present in the webinar of BioTuring. And we are going to do a series of webinar for high impact papers using single cells, and we will send you an announcement later. So I get some questions from the audience. Can you, from Nicola, can you analyze the number of analyzed cells affect the clustering results? So the question is whether the input, the amount of cells actually input the result. Yes, the amount of cell actually impact the results, because, you know, for a given populations, if you subsample, you give us a size of N, and you subsample the populations, and this subsampling should be large enough to represent the population. If it is not large enough, then it can underrepresent the characteristic of the population as well. And also, if you have very small number of cells, also it can affect the scatter plot, because the scatter plot was performed by the Disney algorithm. But essentially, if you have more than 50 or 90 cells, then I think it can represent the populations very well, especially when you work with other SMART-seq protocol, not 10x genomics, but SMART-seq protocol, which has about 96. I think it works well on the systems. Questions that we obtain, and again, I would like to thank Dr. Ankur Sharma and his colleagues for presenting an excellent paper that is, reproducible and has a great impact for understanding the oncology and the drug resistance for cancer treatment. Thank you very much, everyone, and I think we will send everyone the slide as well as the videos, recorded videos. All right. Goodbye, everyone.
Goodbye. Thank you. Thank you for hosting this.